Hello, ladies and gentlemen, and welcome to our YSI webinar entitled How Algae Sensors Work, Principles and Practice in Water Quality Monitoring. Today's webinar is part two of a six-part series concerning how sensors work on water quality monitoring. We've actually scheduled one per week, and each week we have a product specialist join us from YSI to share their knowledge and experience concerning the various sensors. Today's presentation is being presented by Dr. Stephanie Smith, who has a PhD in microbiology from the Ohio State University. For those of you who are not aware, Ohio is also the state where YSI is based in Yellow Springs. Dr. Stephanie Smith is an expert in blue-green algae and their toxins. She's also a product segment manager for the YSI Environmental Solutions. And she has over 20 years working with algae and other photosynthetic microbes. Just a little FYI for our audience today. Stephanie, we're very grateful to Dr. Stephanie for presenting at today's webinar, particularly because due to time zones, it's now 6 a.m. in the morning for her. She woke up at 5:30 to join us today. So a big thanks to Stephanie for that. For those of you who are not familiar with the GoToWebinar system, first of all, welcome. It's not a difficult system to use once you get used to it, and the interface is quite sleek. You'll notice that it's sitting there on the right-hand side of your screen. One particular area that I would like to draw your attention to is the question box. We will be holding a question and answer session at the end of today's webinar. And at any time throughout today's webinar, feel free to just type in your question in that question chat box. All questions will be queued and we will address them at the end of the webinar. If for some reason we can't get to your question due to time constraints, rest assured that we will be following up with the question offline. One other piece of information to share with you is that we are recording this webinar, and a link will be provided to you in a follow-up email that comes about 30 to 60 minutes after the webinar is complete. All right, let's just quickly walk through the agenda for today. We'll be discussing why monitor for algae, the evolution of algae monitoring, how algae sensors work, the principles, and then how algae sensors work, best practices. So first, before we begin the webinar, I'd like just to begin with a poll to get an idea of the type of algae sensors that our audience is interested in. So I will be launching the poll shortly. There you are. So you'll notice there's a question on the screen that says, why are you interested in algae sensors? So we do encourage you to reply. Just simply select any of the answers that apply to your situation. Why are you interested in algae sensors? For academic research, perhaps drinking water source monitoring, concerned about your safety, or are you simply a technology geek? So while we're waiting for our audience to respond, Dr. Stephanie Smith, I invite you onto the stage now. Welcome, and thank you so much for participating in today's webinar.
Oh, thank you so much, Michael. I am so happy to be here with today's audience. Coming to you here from the heartland of America. And I will just let everybody know that's listening, as Michael mentioned, it's very early in the morning here. And believe it or not, my husband is about to leave for work. So you might end up hearing a dog bark. So that's because Earl the coonhound is sad to see John go. probably nobody more sad than John himself to have to go to work. So with that, Michael, I think I'll go ahead and proceed. And I do want to encourage everybody to participate in these polls because it's actually information I sometimes will use to kind of tweak the webinar and see how things are going. So let's first remind people of what I am talking about when I say algae. Algae, like plants, are photosynthetic organisms that can be categorized in a number of ways. Now, what you're looking at here is not really a scientific way to look at algae. It's close, but really it's a convenient way for those of us who do monitoring because we really want to separate out the macroalgae, which are like the red, brown, and green seaweeds, from the microalgae or the single-celled algae. Now, personally, I divide the microalgae even further into the blue-green algae, which if you're a scientist, you know are called cyanobacteria. And occasionally you'll hear me mention them. I think the key thing there is that these are bacteria, not algae, even though they do photosynthesis like algae. And that's why we always call them algae and refer to them as blue-green algae. The eukaryotic algae are those that cause things like red and brown tides, while the blue-green algae cause blooms we commonly think of in drinking water reservoirs and other freshwater systems. So that means that the microalgae, like the macroalgae, are found in both freshwater and marine environments. Now, people monitor for algae for a number of reasons. I always like to start with the fundamental researchers who are studying, the processes listed here, among other things. The main reason I started studying algae years ago was because my dissertation work regarded the enzymes involved in carbon fixation, which is a major part of photosynthesis. Now, as a reminder, taking you back to your school days here, photosynthesis is the process where plants and algae condense carbon dioxide with water, and they use energy from the sun to form the chemical bonds in sugar. They release oxygen in the process. But today, uh, all academic research aside, the reason we most hear about algae is because of drinking water safety. Whether it's Taihu in China or Lake Erie here in the United States, harmful algal blooms are now the most frequent, and they're more frequent and they're more intense than they have ever been. And of course, the big concern is the toxins that they release and whether those toxins can get into drinking water. This has been problematic really around the world. And those toxins do have an impact on animals ranging from, dogs to livestock and yes, humans as well. So people with these different interests come to algae monitoring with very different questions in mind. The scientists might be interested in how a lake switches over from a healthy algae population that includes things like diatoms to unhealthy algae like blue-green algae or cyanobacteria. But a drinking water manager is often more concerned about the quality of the product he's delivering. And by the way, these blooms are also a major contributor to taste and odor issues in public drinking water. Of course, the public may simply want to know, is it safe for me to go to the beach and swim? Probably they figured out the answer is no if they encounter a fish kill like this one in Florida during their red tide last year. But in any case, these different interests have led to very different tools being developed. And I want to talk about the evolution of those tools. But Michael, I'm curious whether our audience has used any of the tools we're about to discuss. So let's see what they have to tell us about that.
So we've just launched a poll, Stephanie. Have you used any of the following monitoring tools for algae? So we do invite our audience to reply. The choices are microscope, chlorophyll extractions, algae sensors, satellite forecasts, or online analyzers? And we're just collecting responses right now. We'll just give a few seconds for the audience to reply. So, Dr. Stephanie, in your opinion, which do you think would be the most popular choice for our audience
Well,
today?
my guess would be microscopy. My hope, which might surprise you, would be the online analyzers. And if any of you aren't sure what we mean by those, we mean things like the FlowCam or the BBE analyzer. But I certainly would be happy if people said sensors, since that's what I'm here to talk about.
All right, well, let's take a look. And microscopy you are correct. It seems to be the most popular choice.
All right. Well, I appreciate people responding because that does help me understand what my audience is learning about today. So let's talk about some of the tools that people might use to study algae. Michael, thank you very much for that poll. Now, one thing I'll remind the audience of is, what I'm showing you today is just the major tools people use. We don't have time to talk about all of them, but I do think it's important to hit on the most common ones that no matter how old these tools are, they still play a role in algae monitoring. So, as many of you know, microscopy is a low-cost technology. It does require considerable skill, and I would mention that people often underestimate this. Today, there are multiple types of microscopes, but it all began in 1674 when Antony van Leeuwenhoek described the blue-green algae Spirogyra. Now, did you guys know that this blue-green algae was the first microbe ever viewed under a microscope? So that's pretty interesting that microscopes, here we are 400 years, 350 years later, maybe, still using microscopes to look at blue-green algae. Now, today, they are still very, very popular because, as I mentioned, they're affordable, and you can tell a lot about the community of algae at a site just using a microscope. Um, it is the most popular tool for counting algae as well. People often want to know how many cells per mil there are. But I always warn users that there can be a wide variability in the enumeration of algae populations using a microscope because analysts often categorize the algae just slightly differently. So having a good species key and standard procedures is really, really important if you're using microscopy. Now, the other thing that might have been a close second is quantifying the pigment chlorophyll. And this is usually done after extraction with an organic solvent of a sample that is captured on a filter such as shown here. Now, this is one of the most popular ways to monitor the relative abundance of algae in the environment. The unit of measurement is a microgram of pigment per liter of sample, or a part per billion. The chlorophyll can be measured on a spectrophotometer, a fluorometer, or even the fluorescence detector of an HPLC. Now, this is one of the most deeply entrenched methods, particularly in the research and regulatory communities. And as we're going to see later, it really does influence how people feel about sensor technologies. For now, I'm just going to say that as scientists, we should all appreciate the role that sample prep plays, not only in this analysis, but in all analytical methods. So one should be really cautious in making comparisons between methods that don't use the same sample preparation. Of course, today we're going to spend most of our time on sensors that are placed directly into the environment for assessing the abundance of algae. The sensors are detecting algal pigments, not the actual algae, and are used to look at changes in relative algal abundance. So sensors are rapidly becoming the most prominent tool for direct monitoring in the environment. You can see here a YSI water quality sonde of the vintage age. This some of you recognize and probably are still using. This is our 6 series sonde. This is the 6600 V2. These technologies, even evolving as they are still most powerful when paired with some of the other methods we're talking about and that I've already shown you. Now, these are also being complemented by very sophisticated hyperspectral and fluorescence measurements on a gross scale using satellite technologies. We won't discuss this much today, but I want to point you to an update that came out last year. You'll get this hyperlink with these slides. This was about the dead zone here in the United States in the Gulf of Mexico, which of course borders the western coast of the state of Florida, shown here. This is where we have red tides here in the US, and satellite technology is being used to predict what the size of a bloom will be. In this case, we've had blooms the size of the state of Massachusetts in the Gulf of Mexico. That's close to the record set in 2018 and way larger than the 5-year average. So these types of predictions for large water bodies like the Gulf of Mexico and even Lake Erie, they're growing in popularity, and they're really useful for regulators and the public. The challenge is that they are still not useful for small systems that a lot of us are using for drinking water reservoirs or studying in other ways. Now, I changed this to say image analyzers because I recently met someone from FlowCam and I realized they don't call them online analyzers. But to me, I think this is the most exciting technology on the market today, which you might be surprised to hear since I don't sell one of these. But this is now becoming a little more mainstream, and this is a technology that really combines the best of all worlds. It pairs fluorescence-based detection with imaging of the algae. So it's like a fluorometer and a microscope in one, along with the camera. I think tools like the FlowCam made by Fluid Imaging Technologies, which started here in the state of Maine in the United States, I think this may be the future of algae monitoring. It appears that I'm not alone in that. Just this month, FlowCam was purchased by the Japanese company Yokogawa Electric. So I suspect that we're going to see some very exciting things happen with this technology in coming years. Hopefully, one of the things that might happen is the price will come down. This is still very expensive technology. It's really not yet well adapted for real-time continuous monitoring the way sensors are. So let's sum up this section. I want to leave you with a number of points. First of all, just because a tool is old doesn't mean it's not useful. And I hope people are that way too now that I think about it. I don't want people to think that microscopes are going to be going away anytime soon because of these newer technologies that are coming on board. The best programs, the best algae monitoring programs use multiple approaches, a combination of these tools, including some things that I didn't even discuss here, like DNA analyses. That is starting to become more popular. So in the future, people will want platform-based technologies that give you more than one type of information. And ideally, you'll get that information in real time. So these include multi-parameter platforms like YSI's exosondes that I'll talk a bit about today, as well as fluid imaging technologies like the FlowCam that I just mentioned. So that was an overview. If you want to hear a little bit more about even some other things, I did write an article about this last year for our Mission Water magazine that we publish. This is available online. You can go here and download any issue of Mission Water, and I really encourage you to do that. This is an amazing magazine, and I wrote a nice little article last year called From Cells to Satellites. So let's move on to the real, as we say in the United States, meat and potatoes of our conversation today. Michael, let's find out a little bit more from the audience about why they are most interested in monitoring for algae.
Great, Stephanie. I've just launched the poll. The question entitles, what types of algae are you most interested in monitoring for? So to our audience, we invite you to answer. Please select one, freshwater blue-green algae, marine blue-green algae, red-brown tide algae. And Stephanie, you're going to have to help me with this last one. I'm not sure if I can pronounce it properly. Macroalgae, is that correct?
That is correct. Well done.
Also known as duckweed, I assume.
That is very good. That is one of the macroalgae, yes. I'm
Okay,
very interested to see the answer to this, Michael, with this international audience. When I do algae talks here in the United States, um, most of the time I'm dealing with folks that are interested in the freshwater blue-green algae, but I wonder if we'll have more marine folks today.
okay, well, let's take a look. I'm sharing the results. Freshwater blue-green algae, 65%.
Wow. Okay, well, that really helps me understand the audience interest, and I'll emphasize that, I think, in this next section because there's a lot of material for us to cover. So, in fact, so much so that I usually divide this section into three subsections, if you will. I want to remind people about some of the fundamental principles of fluorescence. And one of the reasons I do this is because we often forget these things, and this really drives your understanding of what the sensor is going to do for you. And you need to understand that before you make conclusions from using the sensors. So let's go back to school and remind ourselves that a fluorescent molecule is one that can absorb light. We term that excitation, and it emits light, which we call emission. The electromagnetic spectrum is shown here to remind you that shorter wavelengths of light on the blue end of the visible spectrum have more energy than longer wavelengths of light, which are on the red end of the spectrum. So when a fluorescent molecule absorbs light energy, we say that it's excited. Now, that energy can't be held. So the molecule has to return to its natural energy state, and it does that by releasing energy. There are three fates of that energy. Some of it is lost as heat. Some of it might be passed on to another fluorescent molecule, as we're going to see in a moment. And some of it is released as light of a longer wavelength, which means it has less energy. So that release of light is what you will hear me call emission. Now, chlorophyll, that we all know and love, is a fluorescent molecule. It absorbs high energy blue light going to an excited state, and when it returns to its natural stable state, it emits red-orange light. Now, not all of that energy is released as light. Some of it is lost as heat, like we just discussed. But importantly, some of that energy ends up in the chemical bonds of sugar during the process of photosynthesis, which I've shown once again here. Now, that sugar is what fuels the growth of all photosynthetic organisms, including algae. But it ends up that chlorophyll really doesn't work alone. It's not the only pigment involved in photosynthesis, and the energy doesn't go directly to that sugar. It gets handed off to a number of molecules along the way. I could talk all day about that. It is one of my favorite topics, but today we're just going to focus on these pigments. So when we talk about photosynthesis, I want to remind you about why we're talking about that for sensors. When we talk about chlorophyll, the important thing for this audience to remember is that it is found in all of those microalgae. They all use it for photosynthesis, whether we're talking about the cyanobacteria or the Noctiluca that I showed you on an earlier slide, they all have chlorophyll. But chlorophyll is not the only fluorescent pigment, particularly in the blue-green algae. Blue-green algae also have a complex of a pigment called allophycocyanin, which you rarely hear people talk about, and phycocyanin. Uh, these actually are proteinaceous pigments. That's something for if I have any biochemists in the audience, they may find interesting. So the freshwater blue-green algae have very high concentrations, particularly of phycocyanin, which is responsible for the bluish hue some of you have seen when these algae are in the water. That bluish hue is why we call them blue-green algae. But in marine environments, the blue-green algae additionally have another pigment called phycoerythrin, and they have a lot of phycoerythrin. So it ends up that phycoerythrin is a very handy target when we're looking at blue-green algae in marine water. Another reason for that some folks may not be aware of is that phycocyanins, there are different versions of it that can be found in some macro algae. So phycoerythrin is the target pigment for the blue-green algae in marine environments. Now, all together, these pigments form something called the phycobilizome. You can think of this as a light harvesting antennae for light. So these phycobilizomes are integral to the high rates of photosynthesis that blue-green algae can achieve, so that when they have all of their other nutrients, especially phosphorus and nitrogen that we hear so much about in terms of pollution, these algae can grow in a way that often outpaces other types of algae in a system. This is why we very often get that shift from a diatomaceous population of algae over to a blue-green algae. So these fluorescent pigments do all the things we said that fluorescent molecules do. They selectively absorb specific wavelengths of light from the sun. They're going to lose some of that energy as heat because they can't hold on to it forever, and they're going to emit some of that light of a longer wavelength than they absorb. In the phycobilizone, some of the light is released by one pigment and actually gets absorbed by another pigment. So both phycocyanin and phycoerythrin release light that gets absorbed by chlorophyll. So they excite chlorophyll. In this way, these accessory pigments, as we sometimes call these, channel the energy they capture from the sun to the chlorophyll and help drive those high rates of photosynthesis. I almost think of this as like a pigment funnel, just funneling all of that energy to chlorophyll into photosynthesis. Now, algae sensors were built to take advantage of that natural process. So instead of sunlight, an LED in the sensor emits a very specific wavelength of light to excite a specific pigment. A detector is going to see the light that gets emitted by the fluorescent molecule, in this case chlorophyll, and it's going to use filtering approaches to see only the wavelengths of light that are relevant, I should say, to that sensor's purpose. Now, this principle was not invented at YSI, not by a long shot. And it wasn't even invented for sensor technologies. Fluorometers have been doing this in the laboratory for decades. Every fluorometer has a light source, typically a lamp that emits a very wide range of light wavelengths. The fluorometer is going to have an excitation filter that narrows the range of wavelengths to include those that can excite the molecules of interest in your sample. We're talking about chlorophyll in this example. That filtering, though, is not perfect. And if this is an environmental sample or in the water itself, other molecules might be excited by that light. So there's an emission filter, which is going to narrow down all of the light emissions that reach the detector, and it's only going to be the emissions that are specific for the molecule you're trying to detect. That filtering gives you something we call specificity, meaning you are mainly or only going to detect the molecules you're trying to measure with the detector. Now, through the years, fluorometers have also become highly sensitive, meaning you can detect very, very low levels of the molecule in the sample, much lower than ever before possible. Now, this is something we bring together in our sensors at YSI. The challenge is, how do you bring that principle of fluorescence, which is usually done on the benchtop in a lab, into the water? And in something that's small enough to deploy in the water. So YSI's EXO and ProDSS platforms do just that. That's one way that we advanced the technology, was the packaging of that fluorometer into something about the size of a cigar. So at YSI, we have two types of sensors. We have the total algae phycocyanin, or PC sensor, and the total algae phycoerythrin, or the PE sensor. Both of these are available on both platforms. So really there's four sensors, but it's the PC and the PE, either on EXO or ProDSS. So let's talk about how these work. This is a diagram of the total algae sensor for either PC or PE. I'm going to work my way around this diagram, but in the interest of time, I'm going to skip the wet mate and the titanium housing and the sapphire lens, though all of these are very unique features of YSI sensors that make it really rugged in the field. We're going to focus on how we leverage those principles of fluorescence, where specific LEDs and filters allow us to excite the molecules we care about, and a photodetector, which you may hear me call a photodiode, is going to see that light emitted by chlorophyll. You may recognize these are the same basic components of a fluorometer. Now, YSI has dual channel sensors, meaning that each total algae sensor can excite two pigments. Every sensor has an LED to excite chlorophyll. But then there is a phycocyanin sensor, that PC sensor, which has an orange LED, and there is a phycoerythrin sensor, which instead of the orange has a green LED. So remember that phycocyanin is found in most blue-green algae, but is highly abundant in the freshwater algae, and phycoerythrin is highly abundant in the marine blue-green algae. Oh, sorry, I meant to also mention that that photodetector, tying back to our primary principles, picks up the emission side, and these LEDs are what does the excitation. So if you want to connect the dots to what we were talking about with fluorescence, that's how you make that connection. Now, the total algae phycocyanin sensor is by far the most widely used, and judging on that poll result earlier in the webinar, Michael, it sounds like this is probably what this audience will be most interested in as well. So we're going to discuss it first. Again, the two channels are a chlorophyll and a phycocyanin channel. Now, in this sensor, the chlorophyll pigment in the water is going to be excited by a blue filtered light that is put out by the sensor. That excitation band is 470 nanometers plus or minus 15 nanometers. Now, if you were to look at older or competing technologies, you're going to see that the excitation wavelengths have to use a much wider bandwidth around them. This is an improvement that better filtering technologies have brought us today. We have a much tighter bandwidth here. The photodiode, the emission side of the equation, detects the emissions from chlorophyll at peak wavelength of 685 nanometers, plus or minus about 20 nanometers. So that is what the chlorophyll channel acts like on the total algae PC sensor. Let's look at what the phycocyanin channel does. It is likewise excited by a very tight wavelength band, this time in the orange region of the electromagnetic spectrum. But here is where we do something unique. The light emitted by the phycocyanin reaches the detector, but ultimately what we're looking for is the light emitted by chlorophyll. So we're leveraging that natural process where phycocyanin transfers most of that energy to the chlorophyll molecule. And what we're detecting on the emission side with the photodiode is still 685 nanometers plus or minus 20. So you have a different excitation, but the same emission wavelength being detected. This is very unique to YSI sensors, and it's among the reasons that our sensors are so sensitive. We are using the phycobilizome as a signal amplifier, and we only detect the chlorophyll. Now, the total algae PE sensor works pretty much the same way. And since most of this audience is interested in the phycocyanin, we're going to go over this pretty quickly. The main difference is that you'd use a different LED to excite the phycoerythrin. So the chlorophyll channel works exactly as we just discussed, but the phycoerythrin pigment is excited by a green light at about 525 nanometers, again with a very tight bandwidth of 15 nanometers. That energy gets transferred to chlorophyll, and that is the emission that we are picking up with this sensor, just like we did with the total algae PC sensor. So you may be wondering by now how the sensor, if it's only detecting chlorophyll, is telling the difference between chlorophyll and phycocyanin. So we're going to get to that in a moment, but for now, let's talk about the brains of a sensor, and that is its printed circuit board. Every sensor has its own PCB. This is something very interesting and that YSI was one of the first to start doing. I learned working with that R&D team at YSI that the circuitry in the PCB is sort of like a car engine in idle. It's always humming in the background, and it's causing a baseline frequency that we call noise. If the board is not well designed, that car engine is idling higher and the noise is higher. That makes it harder for the sensor to know what the real signal is that's coming through the board via the photodiode. So really good board design leads to good signal to noise ratio, which means you get better, meaning lower detection limits. So getting that really high sensitivity, which is what we mean by low detection limits, without sacrificing the specificity that you got with these LEDs, that is the mark of a great sensor. Now, specificity is further optimized through firmware, which is a fancy word for the software that resides within the sensor. Modulation of optical frequencies means that the sensor has software which enables it to reject noise and out of band frequencies so that the sensors don't interfere with one another when they're sitting on that exosonde. I think of this as the tools used today in the pop music industry where they modulate the frequencies of the singer's voice. So Katy Perry's recorded voice is not her natural or native voice. It is modulated to have perfect pitch and tone. The firmware does that for the sensor. It also aids in what I refer to as electrical filtering. Stray light does reach the photodetector. I'm often asked if ambient light interferes with these optical sensors. It doesn't, and in part that's because of the firmware. So the sensor knows the light that is coming from only the stuff you care about. It knows that also via timing, in a manner of speaking. That's the answer to the question I posed on an earlier slide about how the sensor knows when the signal comes from chlorophyll excitation or excitation of one of the other pigments, phycocyanin or phycoerythrin. The LEDs that excite these pigments are not all firing at the same time, and they are pulsed at specific frequencies. So the firmware allows the sensor to tease out the relationship between the timing of the LED pulses and the timing of the signals that reach the photodetector. Now that's a bit of a complex concept, and obviously there is some proprietary information involved in that, so we don't get to go into it much deeper. But the key point is that every individual sensor is tuned in our factory. This is a third thing that we do. So when the sensors are as sensitive as we are talking about, it becomes almost impossible to make them identical during manufacturing. Bear in mind that at YSI, Every sensor is put together one by extraordinary people we have on our manufacturing floor. So we must manually normalize every individual sensor and how the power flows through those LEDs. This is a principle I'll touch on a little bit in the next section when we talk about the importance of calibration. Now, why does all of that matter? So the point of the last several slides is that good anatomy drives good function. A great deal of thought went into the engineering of these sensors. They were not just an imitation of the old 6 series sensor or other sensors that our competitors still use. That good engineering has given us sensors that drift a lot less, they require a lot less power, they last for years. You get a highly specific signal for the pigments you want to detect, with a lot of that noise filtered out. SNR here means signal to noise ratio. That helps you get very, very high specificity. Now, the last thing I'll point out is that the combination of hardware and firmware also means that the sensors are less prone to environmental interferences. Those of you who have shifted from the 6 series to EXO have probably noticed this. You get a lot less interference from things like turbidity. How does that look, in fact, when you compare the 6 series blue-green algae sensor, as we used to call it back in the day, which measured phycocyanin, compared just to the phycocyanin channel of the EXO? So we've teased this out just to make that comparison. So the tighter bandwidth on EXO means that you get better specificity. And the optics also require less power. That's a good thing because it doesn't drain batteries as fast. A big thing I've seen is that EXO, as I just mentioned, is less prone to interference. And finally, the EXO is 10 times more sensitive, meaning that you're going to get earlier detection of a threatening algae bloom. So to sum up this very dense section, form drives function. And I want everyone to really understand and think about what these sensors are doing when you're using them for your monitoring. It's important to realize that the raw signals have been processed through hardware and firmware to give you your units of measurement, which we're going to talk about in the next section. There's an optimized signal to noise ratio, and exquisite sensitivity has been achieved without sacrificing specificity. Of course, EXO has the most field ruggedized sensors thanks to that titanium body and the wet mate connectors. But that sums up for you how the sensors work. Now, having a great sensor is only as good as the person using it. So if you want to get all those great benefits of that great engineering, you got to use these sensors right. And that is what we are going to talk about next. Michael, why don't we take a little bit of a breather here and find out, now that our audience knows about how these sensors work, I wonder what they think the biggest challenge is when using them.
Terrific, Stephanie. Thank you. The poll has been launched and we now invite the audience to reply. What would you guess is the biggest challenge people face when monitoring for algae? So please select one of the below. They don't properly calibrate the sensors. They don't take measures against sensor fouling, and they don't understand the units of measurement. So Stephanie, we'll just give the audience a few seconds to answer.
Sounds good, Michael. And this is an interesting question because in working with customers, I see issues sometimes with all of these options, but one of them really, really stands out, and it often is not the one people expect.
And which one do you think that is?
Well, usually people think calibration is the biggest issue.
Okay, well, let's stop the poll and share the results and find out if you're right. They don't properly calibrate the sensors. 63% of our audience responded.
Yeah, and that's what I see all the time. And I think sometimes people think that, one, these sensors are not, frankly, very easy to calibrate relative to some of the other sensors. But what I want to remind everybody of, and what's going to become more apparent as we go on here into this next section, is that these are basically sensors that are measuring a trend. So the absolute measurement, well, we'll see. We'll see if that is what is most important here. So let's talk about best practices. And, I've been at this for a while, and that's one of the reasons I was able to anticipate that that would be the most popular answer. I've done a lot of webinars about algae monitoring where I discuss a lot of ways to look at and interpret the data. We all love to talk about the finer details, such as different types of chlorophyll, A versus B, whether the sensors pick them up, temporal and temperature effects in environmental monitoring. That's something people should be aware of. Which pigment channels would give what outputs for an algae bloom? I've covered a lot of these topics, and I touch on some of them in a couple of blogs, including one that I wrote last year just after doing this same presentation. Now, the funny thing is that in practice, some of these high-minded principles are rarely the things that trip users up. Rather, it's usually the most fundamental things that we're going to talk about now, including calibration, fouling, and the units of measurement. So let's carry on and talk about calibration. So I mentioned earlier that sensors are hand built at our factory in Ohio. One of the consequences of that is that the raw voltage output, which is what is modulated by the firmware, cannot be the same for every individual sensor. This is just a function of physics. If we had near perfect manufacturing, physics would always get in the way on this. So that means that every sensor has to be tuned, and we tune it so that there is a scale of 0 to 100% relative fluorescence units. So what we're doing is we're snapping this region to be that 0 to 100% range in the sensor for relative fluorescent units. Now, although that RFU scale cannot be identical between individual sensors at the point of manufacture. And further, as you use a sensor, the output from the sensor is going to drift so that sensors are no longer identical in their output. So we're comparing three sensors here, A, B, and C. All of these sensors are going to be a little different at the point of manufacture and when you get them in the field and as you're using them. There are a number of reasons that these sensors drift, including the age of the LED, which really means how many hours of use the LED has on it, just like that car engine, and how long it's been since you purchased it. The environmental conditions where you're using the sensor can affect drift. The quality of the engineering, power surges, all of these lead to drift. And by the way, I just want to say, beware of vendors who tell you that they have drift-free sensors. A well-engineered sensor surely does drift more slowly than a poorly engineered sensor. But physics are physics, folks, and sensors drift. And if it's an optical sensor, it drifts. So these are the reasons that we calibrate sensors and also why it is prudent to check the sensors to see when they need to be calibrated. So have you ever wondered why YSI doesn't just tell you when to calibrate a sensor? Well, it's because you have to decide how much drift is acceptable for you, and we should not decide that for you. So, how frequently you calibrate really depends on your tolerance limits and all those factors I just mentioned in the last couple of slides. But the important thing to note is that what calibration is doing is it's snapping those lines together so that the data collected with one sensor can be compared to the data collected by another sensor. This is really important if you're using multiple sensors. Some of you are going to hot swap a sensor in the field, or you're going to swap out a sonde, or you have multiple monitoring sites. If you want to compare them, the best practice is to do good calibration. So, what does that mean? And I want to address some language that is used in the industry about calibration. You will hear people, even here at YSI, refer to a 1-point calibration. Sometimes we just say a 1-point cal. Oh, I wish we wouldn't do that. So, in reality, a 1-point calibration is not really a calibration that's going to necessarily give you all the results you want out of these sensors. What a 1-point cal is using a water standard to basically reset the 0 point on a sensor. So if you're using 3 different sensors, sensors A, B, and C in your study, and you do a 1-point cal, they're going to line up pretty well on that low RFU range. But depending on how different they were at the higher end is going to depend on how well they compare when you get into higher RFUs. Now, if you're mainly interested in very early detection and low concentration of algal pigments or algae populations, a 1-point calibration may be just fine for you. If you're working in an environment where readings may surpass, I generally say about 15 to 20 RFUs, I strongly recommend that you do a 2-point calibration. So that is the first thing that I would call a best practice. A 2-point calibration is going to use a second standard further out in the RFU range and snap all those lines into line so that you can compare the readings across the entire RFU scale for all 3 of those sensors, or however many sensors you have. People doing harmful algal bloom monitoring, and it sounds like our audience is mainly interested in freshwater monitoring, especially folks using multiple sensors at multiple sites, or if you're hot swapping, do a 2-point calibration. Now, some other practices that I won't spend time on regard cleaning of the sensors and the tools used during calibration. Now, in the case of EXO, one of the great features is you can do what's called a multi-cal. And I want to thank a friend of mine at Lake Erie, Ed Verhami at Limnotech, shared this picture with me where he's doing what's called a multi-cal. You may recognize the color of this LED, this orange LED. That means these are total algae phycocyanin sensors. They are very clean thanks to that central wiper, but he is doing a multi-cal. He's calibrating all of them at the same time on the same sonde using the same calibration standard. This is going to give him really great alignment of these sensors when he plucks them off of here and puts them onto one of the other 30 or so sondes that he is using. But it ends up, and here's the answer to that poll question, that biofouling is the #1 scourge of algae monitoring and continuous monitoring in general. It costs people a lot of time and money, especially for optical sensors. Now let's quickly review why that matters. So this is really old data. I'm kind of like one of those old professors who sometimes doesn't update her slides, but I like to share it because it really shows how important it is to prevent fouling. So the top panel shows the chlorophyll readings from one of our old 6 series sensors. Now, these sensors had a mechanical wiper on the sensor instead of the central wiper, and these yellow dots are chlorophyll extractions. Remember that method we talked about early in the webinar of samples that were collected at the same site and the same time. Now down here is a sensor from one of our competitors, and they refer to this as a high accuracy chlorophyll sensor. And in fact, this sensor is actually still sold in the market today if you're using some competing technologies. This sensor has been pegged. And what we mean by pegged is that the sensor thinks there is an amount of chlorophyll that is maxing out the range of this sensor. That is because there is growth of algae on the sensor's face. And all of that signal from that biofilm is reaching the photodetector. So this is a misleading outcome that I see all the time. You may hear a little bit more about how important that is when Carrie Hubbard talks about biofouling in her turbidity webinar coming up next. So the EXO platform has a central wiper. It is, by the way, and I'm proud to say this, the best central wiper in the industry. Its efficacy is shown here in a marine deployment. Um, some other things include using copper. So this is a copper sensor guard that helps prevent some of that fouling. As you can see in the middle picture, this was a very high fouling environment, but that sonde guard has almost no growth on it. And then the wiper did a gorgeous job of keeping the faces of those sensors clean. So I'm not going to go much more into that because we just did a great webinar on this topic. My colleagues Carrie Hubbard and Kurt Butler talked about strategies to fight fouling. You've got to watch it. You've got to pick up those strategies. That is the second best practice that I recommend. And even though I'm recommending it second, it probably is the most important thing you can do. Now, it may surprise you that I'm saying this is more important than calibration. Maybe the next thing I talk about will tell you why I feel that way. So the other thing that I consider best practice is to use the right units of measurement. So listed here are units that are options on a number of sensor technologies, including at YSI. Now, hindsight being what it is, I wish our industry had never tried to translate these lab-based measurements of parts per billion of pigment, cells per mil, and God help us, colony forming units per mil to monitoring tools. And now that you know how these algae sensors work, it might be becoming clear to you that you can't really measure these things directly in the water. So we're going to talk a little bit about them, and then we'll come back to what I think you should be using, which is a relative fluorescence unit. By the way, we won't talk much about raw units. You can get those units on your sonde. Folks, don't use them. They're really there for the diagnostic people working in technical support and in our repair center. These raw units are not very meaningful for the end user. So part of the reason that these units are still employed, though, the reason your sonde can tell you parts per billion of pigment is because we're all just so connected still to these older technologies where we were getting cells per mil and parts per billion of pigment. That's why people are comfortable with these units. They've been around forever, and we like to think in these terms because sometimes we're trying to compare sensor data to older data. But the sensors really don't directly measure these things. In the field, a sample is collected and it's filtered to grab the algae. And then we extract that with acetone or methanol, depending on the method you're using. And then we may visualize that on a fluorometer like we discussed, or an HPLC equipped with a fluorescence detector. That's all great, but a sensor, when we developed those units to deliver with the sonde, we did it in a lab. So when the parts per billion unit was developed, it was done with a laboratory grown culture that is diluted in a laboratory medium. This is grown with a constant intensity of light on a diurnal cycle, and then we perform that extraction. We measure RFU with the sonde, and then we measure it in a fluorometer and we build a correlation. That's how we deliver that unit to you. However, all of you that are microbiologists know that what happens in the lab stays in the lab. It's really not representative of what goes on in the environment at all, and a lab grown culture doesn't act the same way. So in some cases, the parts per billion delivered by a sensor may align really, really nicely with what you would get from a chlorophyll extraction. I'd estimate about 60% of the time it does great, but other times it doesn't do great. And so sometimes that comparison is not very strong. So I strongly advise that if you plan to use the part per billion unit delivered by an optical sensor, whether it's a YSI optical sensor or anybody else's, you ground truth that measurement and make sure that it really does align well with the samples from your environment. In fact, that's exactly what my friend James Chen did. James works in China. James is one of our fantastic application engineers there in our Beijing office. And he did this for a lake he was studying. What he did was he collected his own samples, he prepared his own dilutions, and then measured them with the sonde. And then he continued on through the extractions and used a benchtop fluorometer to get that parts per billion. Now, in his case, the results were spot on. The chlorophyll delivered parts per billion delivered by the sonde on the x-axis lined up beautifully with what he got from his field samples. You need to do this because this may not be the case for your samples. Now, there are a lot of reasons for this. I've talked about this in a webinar that's now coming up on a couple of years old. I strongly advise that you listen to this webinar if you need to continue using part per billion of pigment in the samples that are delivered to you by the sonde. Now, there are a lot of physiological examples, reasons rooted in the algae themselves, which can affect the reliability of laboratory units in an environmental monitoring context. This is especially true for those of you that still want to use cells per mil. The exosonde does not deliver cells per mil. This is why. It's because it's really not it's not something we can really build a great correlation on. We did build something into the software that allows you to do it, but frankly speaking, I don't advise it. So we're running out of time, so I'm going to sum up this last point by saying, please use relative fluorescence units. People doing monitoring should be looking for changes from a baseline, fluorescence in the water, not comparability to laboratory analyses. So using the monitoring data will help you decide if or when you should collect samples for your lab. The relative fluorescent unit is the truest measurement delivered by the sensor. So summing it up, calibration, fouling, and using the right units are the three best practices that I recommend over and over and over, and that I think are most important for harmful algal bloom monitoring. RFUs will be most powerful when you build an understanding of your system, like I described in that 2018 webinar that I hope you guys will go back and check out. But today we're talking about how the algae sensors work, so we will wrap it up there. Michael, I hope we have some great questions. We typically do from this webinar, so I'm going to hand it back to you.
Thank you, Stephanie. That was fantastic and very informative. Based on the number of attendees still on, obviously they found it as likewise the same. We still have over 90 people on, and I realize that they are probably interested in the Q&A session. Just to start though, we would like to understand whether or not you'd like additional support from our YSI and regional Xylem staff. So if you could kindly enter your answer to the screen, to the poll question that's currently on the screen. Do you want someone from YSI to contact you to discuss algae sensors? And we will certainly follow up with you over the course of the week. So feel free to enter your reply there. As I said, someone from Xylem will certainly follow up with you. Thank you very much. And I don't want to take up too much time from our Q&A session. I do realize that we've now hit the hour mark, and we will stay on for a few more minutes to take your questions. And once again, please use the question box that's on the side of your screen. Simply type in your question into the chat box. We can see your questions here on our screen, and we will pass those on to Stephanie. While we're waiting for the questions to roll in, you're definitely free to contact either Stephanie or our regional staff here in the APAC area by using the email addresses that are currently displayed on the screen. We'd also like to share with you the the third part of our 6-part YSI sensor series webinar that will be held next week from June 2nd. In fact, all of the webinars will be held every Tuesday at the same time. So it's very easy to remember. If you're not sure where to register, simply go to our Xylem Analytics.Asia website, and it's easy to navigate to the registration page. All right, Stephanie, let's jump into these questions. There are quite a few that have been queued up for you. Let's start with one that came in quite early during your presentation, and it concerns aquaculture. Is there a utility for algae sensors? What utility do algae sensors have for aquaculture? And could you provide some examples?
Yes, that is a wonderful question. In fact, I'll answer it by providing exactly an example. I, many years ago, was invited to come visit a open water offshore site that had open water net pens where they were culturing salmon. And these net pens, I won't reveal precisely where they were for the propriety of the organization involved. These net pens were kind of at the base of a region where a river fed into this coastal region. And that river was draining a watershed that had a lot of microcystis contamination, and that microcystis was making the toxin microcystin. And so there was the concern about whether those harmful blue-green algae, which are freshwater algae, were getting into these net pens and potentially getting into the fish themselves and might affect the food chain. So that was a case where they wanted to monitor the influx from a river of blue-green algae that normally would not have been present. Now, it ends up, but in case you're wondering about this, you aquaculture folks, those toxins don't really biomagnify in the tissues of the fish. So it ended up not being as much of a health concern as they had thought. However, it was a very important thing for them to monitor because the other stuff coming in with the algae was the nutrients, which had a huge impact on how they were culturing. So that is one example. Now, in closed pond monitoring, we do have people using algae sensors, and it depends on where those ponds are and how they're being managed and the type of algae that people are concerned about. So the short answer, which I should have delivered first, is that yes, the sensors definitely have application for the aquaculture industry, and we do have people using these sensors in multiple aquaculture applications today, Michael.
Excellent. Thank you, Stephanie. Another question concerns potential issue of strong winds that are generating abnormal data. Do you believe it's the strong winds that are creating this issue, or could there be some other cause?
Well, that's a good question. Strong winds, if I had to guess, and obviously I don't have a lot of information to work on here, there could be a few effects of the winds when it comes to algae monitoring. And it does depend a little bit on what you mean by abnormal data. But I'm going to assume what you mean is fluctuations in your RFU readings. Most definitely, strong winds and currents move the algae. And so I've seen this here in the American Midwest, where you'll have a scum of microcystis sitting on top of the lake, and just a few minutes later, it's pushed over into a cove because there's a prevailing wind that, a southwesterly wind or something that is just moving that algae. And that definitely can cause spikes in your data. Frankly, though, that's one of the reasons you do algae monitoring, is to see that. Now, the other thing that can happen is, depending on the time of day and those winds, the algae themselves will sometimes modulate their position in the water column. Some of these algae can move up and down in the water column. Either they have gas vacuoles to help them move, or they're flagellated. So winds can impact how the algae themselves behave, which would cause changes in your baseline monitoring. Now, the last thing I'll mention, and I'm not sure if this might be what the person asking the question means, is you might test your power at your site. So sometimes high winds are going to cause interruptions in data simply due to power. So if that is the type of thing you're seeing, where you're just not, all of a sudden your data are dropping off and then returning, that would probably be a power issue, Michael.
Okay, great, Stephanie. I'll move quickly to the next question. Is it correct to assume that the tau PC sensor can only be used for fresh water and the tau PE for marine water?
That is a fantastic question. And the answer is that it is not always correct to assume that. Often that is the case. So probably 80% of the time that is the case. But let's go back to that example that I just talked about, where they were doing those offshore net pens. Those net pens were in a marine environment. And most people in a marine environment are going to use the total algae PE sensor because they're interested in algae, blue-green algae that grow in the marine environment. However, in that particular application, the total algae PC sensor was actually the one I recommended, because the algae they wanted to detect originated in that river that was flowing into that net pen area. So they wanted to detect a freshwater blue-green algae in their marine environment. So I actually recommended that they use the total algae PC sensor in that application. And I have on occasion recommended that. So now that you guys know how the sensors work and what the sensor is really detecting, you can make a more informed decision. You don't have to just rigidly say, always use the PE sensor in saltwater and always use the PC sensor in freshwater. You can make a more informed decision now that you know what the sensor is doing.
Great, thanks, Stephanie. Just a quick note to Kieran. Kieran, you're still on. You were asking about whether or not you can attend the previous webinars. What we will do, Kieran, is our staff will send you a link to the recording and you can listen to it at your own convenience. View on demand, a definite benefit of these webinars. Moving on to the next question, Stephanie. Is there a sensor that differentiates toxigenic to non-toxigenic cyanobacteria?
Oh, thank you, whoever asked that question, because that is a very common misperception. We always talk about these sensors in the context of harmful algal bloom monitoring. And harmful algal bloom typically means toxic algal blooms. The sensor does not tell you anything about toxicity. The sensor tells you that there is the potential for toxicity. I have been working with harmful algal blooms for many, many years, and I have seen microcystis blooms, and I've collected the algae and I've taken them to the lab, and they weren't making even the tiniest bit of the toxin. So just because you have the algae there does not mean you have the toxin. And that's why these tools, all algae monitoring tools, need to be paired with the other analyses that are important to you. So what I tell people is, use the sensor to tell you when you need to collect a sample, and then take that sample back to the lab immediately or freeze it, and do your ELISAs or your extractions in HPLC, whatever method it is you're using to detect the toxin in the lab. This only tells you that there's the potential for the toxin to be there. That's a great question, Michael. Thanks.
Thank you. Stephanie, we've actually received quite a few questions about calibration. So let's select one of them. Which calibration solution should we use for chlorophyll calibration?
You should use rhodamine. So the EXO manual describes how to do a calibration using rhodamine. So you use water for the 0 point, and then you use a solution of 625 parts per billion of rhodamine for that farther out point that I talked about in the webinar. Now, why are we using rhodamine instead of chlorophyll? There are a few different reasons. One is you can make a stable enough solution of rhodamine to do the calibration. When you dilute a chlorophyll to those very dilute quantities, it starts to fall apart. It changes while you're looking at it. So it's very hard to get a good stable solution that is so dilute of chlorophyll. The other reason is that chlorophyll typically is extracted in an organic solvent, and you can't put the sensor in an organic solvent. So that's another reason that you use rhodamine, because you solubilize the rhodamine in water. Finally, the rhodamine is it's relatively cheap. So we recommend one particular manufacturer of a very nice analytically pure preparation of rhodamine. So rhodamine is what we recommend. Now, some of my more biochemically savvy customers have come up with ways to make preparations of chlorophyll, and they in water. It's very difficult to do. You can do it for phycocyanin as well. But generally speaking, I don't recommend it because it's you have to be a pretty good laboratorian to pull it off. Now, if you are, you can try that. There's nothing preventing you from trying that, and you can put in your own parts per billion units during that calibration. But really, rhodamine, it's tried and true. It's easy, it's cheap, it's the best way to go.
Great. Okay. Stephanie, thank you so much. We actually have quite a few questions here, and I don't think it'll be possible to cover them all today, but rest assured to our audience, again, Stephanie and our team will definitely contact you directly with an answer offline. So thank you to everyone who joined. The audience has just been fantastic. A special thanks to you, Dr. Stephanie Smith. Thank you for your time, especially so early in the morning.
Oh, I'm so happy to be here, and we will definitely get to those questions. And I want to thank everybody for sending them in. It is my priority that you guys get the best use out of this technology. So I appreciate your questions because we're going to make sure you do that.
Terrific. Thanks, Dr. Stephanie Smith. And thank you again to our audience. And please join us next week for how turbidity sensors work. Stay safe and have a great day. Goodbye.